Wendy Hartsock, science and peptide enthusiast. In this episode of Exploration Science, Doctors Christian Behn of CEM and Ronald Frank of Ames Scientific Products discuss the development and applications of spot synthesis for peptide libraries. How do you actually know each other?
We know each other since the beginning 90s, Ronald, I would say. When Ronald did the, basically the first presentation of the spot method. I got a new job in Germany at that time, which were involved in automation. And I moved then to Braunschweig. And so Ronald working with the spot method. So that was basically using a standard pipette, putting droplets onto a membrane. I was doing the proofreading, Ronald was pipetting.
Yeah.
I think we did a 5x5 sheet of peptides just to get involved and learn the method. And then, of course, there was a quick decision to do some kind of automation in this direction. And the first automated system was then developed in the mid-90s with software, which was just the error-free dispensing of the activated amino acid onto the membranes. And all the other steps like FMOC deprotection, washings, cappings have been done manually. So it was kind of a semi-automated approach in the mid-90s.
So, Dr. Frank, so you were already doing spot synthesis when you met Christian. You had already developed it. Could you tell me a little bit about how you got interested in spot and then about the technique itself?
Well, I mean, it started quite out a decade before when I was still working on oligonucleotide synthesis. And I discovered that cellulose is a quite practical support for synthesizing many oligonucleotide sequences at the same time by just using disk slides with numbers and then just putting them together as the sequence is required. And so this started out in the very early 80s. And of course, the principle of this method was more general. So I thought peptide synthesis should be also adapted to this. And I went to a lab of Bernd Gutte in Zurich and learned how to make peptides. And when I returned to Braunschweig, I just adapted this to cellulose support. But I mean, in that format, with this disk of some 5 centimeters or a few centimeters diameter, we synthesized too much material than the people used in biological sciences. So the miniaturization was very needed. But then the disks get really difficult to handle and they get smaller and smaller. And then it came up the idea to leave them in this array of spots on a continuous sheet. So this was the kind of very practical solution for this problem. And still it was connected with a lot of manual work. And then at that time I had also quite good contact to Heinrich Gausepol, who was working at Imbel in Heidelberg, and then with a company called Abimed, and they were distributing a peptide synthesizer that they developed in Heidelberg, and this one was very easily to adapt to hold membranes and use the needle of distribution reagents to just spot on this sheet. Mm-hmm. And so we came into contact with Abimed, a company which was kind of predecessor of Identifies.
And so what's the scale of synthesis on spot?
Well, this is quite flexible. I mean, you see that there are membrane types of cellulose which are very thin and very thick. So the amount of support material per square centimeter can vary quite much. And so then, of course, the degree of modifying it with a suitable linker also can vary. So I think we can manage from a few femtomole of a peptide up to low milligram amounts. So it is good for analytical experiments as well as for preparative ones.
And the analytical experiments, so do you have, what do people typically doing? Do they have, how do you get the peptides off the cellulose membrane to analyze them, or do you?
Oh yeah, you can. I mean, in many applications, you don't care about. You rely on the chemistry and do it well. So the array which you produce is directly used in an experiment. But if you want, you can punch out the spot, and in some cases, of course, you have to provide a special linker between the peptide and the cellulose that can be cleaved chemically. So this can be a simple ester bond or a more sophisticated one. But then you can cleave with chemically the peptide from the support and make analysis, amino acid analysis, MS, and so on.
Sure. And do people do a lot of photochemistry with these types of platforms?
There have been a parallel development, which was some kind of lithographic chemistry, but this was a little bit more difficult to handle in the lab. And the most important point at that time, I mean, this started already in the 80s with the pin technology of Mario Geis, and so this was also a 96, I would say, Welle or pin format, or the T-bag method from Houghton, where they needed more compounds and more products instead of just a large amount. They wanted a smaller amount to do kind of a screening, and then of course the membrane became ideal for this one because, as Ronald mentioned, you have a surface. The membrane is stable during the complete synthesis process against the organic solvents and all the reaction conditions. And the purity at that point, I mean, this was, of course, a long time, a learning process for the chemist. I mean, for a biologist, it's clear biology talks first. So we had faith in the synthesis method. So we were aware of the fact that there will be definitely peptides in the single spot. And since we were doing generally short peptides, up to 15 or 20 meres, we know there will be enough material and enough purity to do the biological assay. And this was the only way to test in parallel different compounds. In this respect, different peptides. And then there became several approaches to analyze proteins, to screen proteins. So that leads really in this direction to understand the nature of a protein just by studying the peptides.
Yeah, can you elaborate on that, Dr. Frank, a little bit? Maybe some of the applications that are most utilized?
Yeah, I mean, as I mentioned, the idea came from the use of peptides in immunology. So this was a large demand also at our institute where I worked at that time. And immunological assays are really, really sensitive. So a few microgram was a material where you can work a year with. So a method should be adaptable to this type of analysis. And therefore, I work closely with immunologists and learned from them how they do their assays and how easy it was to adapt this to the membrane where the peptides are already sitting in nice order. So you don't have to transfer from the membrane into a microtiter plate, which the people are also quite widely used in immunology research. So it's more the format that biologists use for transferring proteins from a gel or from a dot onto a membrane. And well, but a format that they're really well acquainted with. And therefore, I think also kind of misinformation is somehow found in some review articles where people just report on literature and never had a membrane of sports peptide in their hand. And
Right.
this is the reference that the membrane type people use is nitrocellulose. But this is totally rubbish, because nitrocellulose, everyone knows, easily dissolves inorganic solvents to generate celluloid. So I hope very much that this misinformation in some of the reviews will not trigger some really disappointing experiences in some labs.
So I guess the confusion maybe comes, nitrocellulose, is that used in Western blotting? Is that why?
It's more common and more used. That's right.
So what are the, so you mentioned cellulose, but you can use other types of membranes and even you call it paper. So what are the different types of membranes you can use?
Well, we started with ordinary chromatography paper, which worked quite well, but we experienced that there are some sequences where you have difficulties to cleave off the protecting groups at the end. So therefore, longer treatment with really acidic conditions at the end to remove the protecting groups became necessary. And therefore, we started a kind of program to develop a more acid-stable membrane, which we now call ACS01. And this, in comparison to normal chromatography paper, is much more stable. So it allows you to treat the membrane with the peptides with classical 80% trifluoric acid cocktail even overnight, and the membrane still survives and can be used in further experiments. So it's a strong recommendation to use really acid-stable membranes.
Right. So maybe, sorry, just thinking about how the utility of spot synthesis, can you compare and contrast doing it manually versus doing it automated? Maybe just walk through the procedure.
I mean, the real biggest point in terms of doing it automated is, of course, to handle the sequences. Let's say you have a sheet of paper we are using our standard setup. This is a 10 by 15 centimeter sheet of paper. You can bring up to 600 spots onto this paper, and 600 spots, and generally you have a 15 mer. So you can count, these are 9,000 spots, double coupling 18,000 spots. None of your technicians would like to do this manually, right? And they have to do it error-free. So the handling of the sequences, and then of course we generated software tools to generate sequences for epitope mapping or for two-position libraries. Then we have other approaches like the alanine walk to understand the nature of certain amino acids or where the binding is. We can do N and C terminal truncation. So this is basically just text files which are organized and handled by the software to generate peptide sequences. And the information which are coming out could be important because you understand which amino acid in this particular epitope is really important by the alanine walk. When you do N and C terminal truncation, you get the idea how many amino acids are important for the binding site. So the automation, the fully automation is not just handling the pipetting and liquid handling, it's also the complete handling of the enormous amount of sequences. And we are just doing a small number. I mean, this was always in our intention to have a smaller number compared to the competition. They can do 10 or 15,000 spots on the slides. This has never been our intention. We want to be free with handling our projects and work with certain areas of the protein, because we can't handle the protein in these kind of assay, but we can handle overlapping peptides. And with overlapping peptides, we can understand the nature of the protein and the binding site. And therefore, we needed a computer program to handle this, an X, Y, and Z pipetting robot to do all the dispensing, because you would get tired of handling the pipettes, right? So automation was really necessary at that time.
Right. Yeah. And so what is so you said 600, 600 spots. So do most people do their peptides in triplicate? Like, what is the standard setup that people like to have on those? Because this is mostly biologists.
On the membrane they can handle different setups, because the work area, you can freely define the array. So very often a 10 by 15 sheet would need a large volume in the assay, 15, 20 milliliters. That's far too much for little mice. So very, very often you do subsets of the peptides, and you can do this in duplicates or triplicates. And then you can cut the membrane in different smaller pieces, and you do the assay. That's what normally people are doing. You can do also a full membrane if you, let's say you want to do some substitution analysis where you have a sequence of a peptide and you want to replace every position, every amino acid by the 20 or 19 natural amino acids. You easily generate a set of 400 or 500 peptides, and this can be screened. And the nice thing is you screen them in parallel, right? You don't work with individual peptides. In a solution phase assay, you have a sheet of membrane, and not to be afraid of this. I always tell it's just washing a sheet of paper. And that's what most of the biologists know and can easily do, because they know this from all these kind of dot plot techniques to handle these kind of membranes. And that makes it so attractive to the system. And I really must say, for the biology, I mean, I'm a chemist myself, but since I'm working now for more than almost 30 years with spot method and with these kind of technology, a single molecule inside the spot will be detected. And that makes it really appealing. You don't have to think about any workup, any purification step, because this is to find the needle in the haystack. This is the first point. You get out of a protein with 500-600 amino acids, you get a small area of a 15-mer peptide, maybe, the epitope is 5 amino acids long, and you work with this particular peptide. And this is easy to do for most of them. And then after finding the sequence with the spot technology, you can step up to a resin synthesis and synthesize a larger amount on resin.
Yeah. So, okay, Dr. Frank, were you gonna?
Yeah, I fully support this, we also have to consider that there is not a growing need for synthesizing really thousands of peptide fragments for one certain project that the biologist is interested in, because with the readily available sequence information now for many model organisms, for human sequences, you always have a kind of blueprint available, so you don't have to fish in the dark to find something within billions or millions of sequences. So therefore, the format of the spot synthesis with some up to 1,000 sequences is fully good for most of the projects.
So along those lines, I think it's also important, some of the references that you had sent me go beyond binding. I mean, these are applicable for enzymatic assays, for even cells, where you're actually putting cells on the membrane. So to your point, Christian, that this is just a piece of paper. So maybe, Dr. Frank, could you talk a little bit about, actually, let me back up and ask you one question first. Can you talk about what you do now as far as AIMS research and what you actually, the services that you provide, and then talk about what are the top requests that you get. And then maybe Christian, you can go into sort of what you're seeing people doing that you find to be most interesting. So Dr. Frank, we'll start with you.
Of course. I mean, AIMS is a company which was founded by myself and the postdoc of the group in 1989. And it focused on more the chemistry on material surfaces. So it was not a biological application company which exploits spots, but it was a more material production company. And so we developed and distributed these more specialized membranes for different projects. And even beyond, we modified for customers certain surfaces of plastic or so, which was needed for their projects. So AIMS is really not an application company. So my part then became, after I retired now 6 years ago, is following the literature on spots. And it's really fun to see that even after 30 years that the method exists, still people are inventing novel applications. And so it gives an impression that this method is still really alive. And as I mentioned, it started all with some immunological research, where the proteins we worked with were antibodies or even T cells. But this antibody-antigen recognition is more a special case for protein-protein interaction. So spots is really usable for any project on studying protein-protein interactions. And this is still a very up-to-date topic. I mean, all proteomic and genomic research tries to find out how the components of the cell are interacting to contribute to the life cycle of the cell. And so proteomics approaches are increasingly using spot peptide arrays to fish out proteins from full cell extracts onto spots for peptide-specific spots to find out how these small linear recognition motifs contribute to building larger protein multi-protein complexes.
Christian, maybe you could talk to about sort of just some of the more interesting applications that you've seen, or some highlights of.
I mean, the highlights are definitely from the beginning, the antibody. So customers are using the membranes also as a quality control sheet for produced antibodies. I see new publications also, especially with the SARS coronavirus. They have used these. You know, they have to identify spike proteins on the cells. So this can be used as well to see the binding site to certain antibodies. Then there's users that are doing antimicrobial peptides, which is really important when you think about people who get allergic reactions. We have a group in Germany, they test sera against peanuts, soybeans, so they want to understand the nature of these parts, and everything, especially the one with the immune reaction, is going into can go into kind of a drug development of vaccines. So as we have seen this with the SARS and Corona, and especially when I heard certain people in Germany, they say, "Oh, the vaccine has been developed so fast." I mean, that's not true. I mean, I have a nice publication from a Canadian group from 2004. They have already detected SARS and MERS at this time, and they were aware of spike proteins and peptides, which could be used to develop kind of an antibody concept there. So there's a lot of research, and parallel to this is the spot synthesis, or put it in this way, the multiple synthesis has been always part of it.
Yeah.
Right.
And sorry, one thing that I saw come up was this, what is microspot SC squared?
Those were,
Shall I comment on this?
yeah.
Okay, so yeah, the development of CS square or microspot was kind of request from biologists to carry out multiple experiments with one defined set of peptides. So, but spots generates with quite amount of work and effort one array with the one set of peptides, which you can use several times, but one after the other. So it is not prone to handle hundreds of different assays at the same time with exactly the same type of assay. So we thought, how can we solve this to limit the amount of work for the synthesis, but to allow a multiple screening of the same set? And then we came up with the idea to punch out the spot on the cellulose and just dissolve the cellulose support, make it soluble so that you can aliquot this and pipette this with in a very nanoscale format onto glass plates, where the cellulose sticks to the glass plate and then thereby immobilizes the peptide. And this can be much further miniaturized down to nano scale. And as one spot generates a milliliter of a solution, which can be spotted thousands of times on glass plates, you can copy this very easily and then use it in a multiple screening experiment.
Yeah, this development, Wendy, has directly lead to our cellulose spot synthesis.
Okay.
And the idea here is, of course, as Ronald mentioned already, you have different patient, different sera, but you want to use the same set of array in the assay. And this is the beauty of the cellulose spot, besides the miniaturization. Also techniques which are known from the DNA and RNA arrays. You can use cover slips to have very small compartments with your assay solution, and that you have duplicates, and you can do copying, basically reprints from one synthesis of up to thousands of slides. Makes it really beautiful, especially if you have a lot of patient and sera from patients to test.
Right. Maybe can you, Christian, just for people who maybe aren't familiar, just walk through the whole process of automating this. I know you already talked about like the programming and the benefits of automated synthesis, but actually like talk about where do you start? And then if you want to get to this microarray printing of slides, where you end up, just walk through that.
Okay, you need one of our CEM fully automated peptide synthesizer, the one for the cellulose spot synthesis. So you have a frame of 384 disks, and you can have up to 4 frames on the machine. So you synthesize in the same way as you would synthesize a peptide on resin, in the same way with the mild F-MOC chemistry, you synthesize your peptides on the cellulose disks. After the synthesis, I mean, Ronald mentioned that in the beginning we were using the full sheet, and after the synthesis we were punching it out. Now we have, as I said, a 384 frame with cellulose disc inside. After the synthesis, we do the standard TFA workup with a little trick to dissolve the cellulose slides. Then you get a 384 well plate, the mother plate, and you can spot these onto glass slides. Onto glass slides, you get a 3-dimensional structure of the cellulose fiber with peptides attached to it. So we get more kind of a 3-dimensional structure. I mean, you know, protein are not linear chains. This has a 3-dimensional structure. And this is nicely represented on the cellulose spots. And of course, you need an arrayer or the slide spotting robot to do the slides. And then you can print on our machine, you can print up to 30 slides. And from the assay, from the cleavage solution, you can do copies of up to 1,000 slides. So you can always do a fresh array copy of the one synthesis you have done, and you get for a year, you get always the same array, and you can test different sera. So it is, one part is a synthesis on a standard pipette, on a standard peptide synthesizer. And then the second part is a workup, the procedure to dissolve the cellulose spots, and then they get printed using a standard array or our machine to print these onto slides. And then you have duplicates on the slide of the 384 set, and this can be then used and tested. And
Awesome, thank you. Yeah.
in a short way.
So what are some of the other molecules that, I mean, so, you know, Dr. Frank, you mentioned you actually started out looking at oligonucleotide synthesis. What are some of the other molecules that are being studied using SPOT?
Well, I mean, first of all, there's a lot of interesting chemistry you can do by introducing the normal biologically relevant post-translational modifications like phosphate, sulfate, acetate, and so on. So a lot of opportunities to modify the peptides chemically. And so then when you leave the peptide scaffold, you could think of oligonucleotide synthesis, but this needs much more closed inert reaction conditions. So this is really difficult because the chemistry is very sensitive to moisture and so on. So there were no real reports, as I know, dealing with oligonucleotide synthesis. But quite early on, people applied this to synthesize small molecules. So using a series of small chemical reactions which are compatible with moisture in the air and so on, and then build up hydrogenes or small organic scaffolds to screen as drug-like molecules. So this are quite many reports and quite nice chemistry that was developed to be compatible with the spotting method. And well, the most recent report I found was that even people try to adapt oligosaccharide synthesis to paper support, which, however, is a very early report. So only very few, on a very small array of oligosaccharides was produced, and which has to cope with, similarly with very complicated conditions like temperature control reactions and so on. So let's see how this will develop. But a very nice and innovative method was developed from a group here in Berlin. They used a kind of collection of drug-like molecules, so several 10,000s there available for screening for drug-like molecules, and they spotted these on a special cellulose membrane which was first derivatized with a photoreactive molecule. So when they spotted these compounds and shine UV light on it, the cross-linker attaches these small molecules in a very unspecific way to the cellulose. And then they incubated this with cell extract and analyzed the proteins that are captured from these extracts by classical proteomics MS technology. And so they really rapidly could identify the known targets of some natural product drugs. So it really works, and I'm really curious to see how this can be used to identify novel targets of even known drugs.
Christian, Dr. Frank mentioned that, obviously oligonucleotide synthesis is very difficult, and because you need that inert atmosphere. But what about PNA synthesis? I know that that's been used obviously like on the automated synthesizers, but what about SPOT?
You made your homework, Wendy, right? DNAs have been used as well, and also successfully used on the spot membrane, because especially in the beginning, since the monomer derivatives have been so expensive, they could only do a very small scale. And of course, attractive has been also the immobilization to the membrane, so they have done these. And this is, I would like to emphasize this. It's the beauty of the full chemistry. You can do post-translational modification with different building blocks, diamino acids, phosphorylated, sulfatated, methylated ones. And you can do this on certain points. It's not just in the end of the synthesis and you get all of it. No, you can have dedicated areas where you can make changes to the sequence. And of course, when you look into the literature, when you want to make a peptide suitable as a therapeutic agent, you need to stabilize these, and this can be done by pegylation, by using different kind of building blocks, diamino acids. You do cyclization to make them more and more stable, and this can be all done in the full. If you do the full chemistry, right? This is not what the biology necessarily can do directly, but a chemist can do this to a certain point. I mean, we are limited in terms of the length of the peptide. I mean, the purity drops, of course, if you don't take any precaution to keep them, to get the running and get always the high yield per individual cycle.
Mm-hmm.
But there is a lot of opportunities in the chemical synthesis, and as Ronald mentioned, they have used also other building blocks, and especially pharmaceutical companies. They have millions of different compounds they want to test, and this can't be done in a large scale. So there needs to find a way in a multiple synthesis way and test them in parallel. And this makes it so. Makes it really beautiful to have these kind of ways with the spot synthesis and the cellulose spot synthesis.
So you did mention a limitation of the technique is the length of the peptides, and that is a result of purity. Are there any groups that are working on like ligation methods or anything? I mean, if these are amenable to, because it's paper, if it's amenable to biology, can you do things like ligation with like NZ-Tap?
I'm not really into it, but I've seen an article where they have used chemical ligation,
okay.
Native chemical ligation on the membranes as well. And of course, this was a step forward in native chemical ligation to do really proteins. I mean, go above the 100 amino acids. But I would say on a parallel synthesizer, you are living in the world where you have to go for the one which is the most difficult. There will be, and if you look onto the synthesizer, especially in the beginning where we had one resin, you could really see by a shrinking resin a bad synthesis and cycle, by the expanding resin a good synthesis, a good peptide in the end. And therefore we have, in the 90s, we definitely have said the limit will be for good peptides without any purification. For crude peptides, this will be up to 20, 25 amino acid. But nowadays, we know this with the microwave synthesizer, with chemical aeration, with pseudoprolines. There's different ways to go beyond. But this you do with this for individual single peptides, and that makes sense to a certain point. But if you want to screen a protein, there is no better way than the small scale on the membrane to do the first step. And then you have the area, and the area is important, and you have the sequence from this area, and then you can step up to larger scale and other assays, and of course, important chemical evaluation of the peptide.
Yeah. Well, let's see, was there anything that I didn't touch on that maybe you wanted to talk a little bit more in detail about?
I mean, the pity at the moment is that I have the feeling that a lot of scientists are not really aware what is available as a method. And this is where we are fighting and working against. So we need to bring the method more into the scientific community, how easy it is. And you don't have to be afraid to do the chemistry of peptides, especially if you do the spot synthesis, right? Most of the machine will do this. We have the mild FMO chemistry, which can be established in most of all the labs. So there's not these scaring HF apparatus where we started in the 60s with, right? So how important it is to understand the nature of proteins and to understand the binding sites, to understand the function of the cell. This is important to bring. I mean, most of the scientists are aware of this, but they are not aware of a chemical process to handle this and do different kind of modifications on the peptide, even post-translational modifications, or study PNAs, study other peptides. I mean, we need to work in this direction of drug development, especially for the peptides, even if there is just 5 to 10% of the whole market. But we have diseases where we need a peptide to overcome, to get the binding. I mean, a small molecule is just the key in the lock. But a peptide can do much more, or can do the most important point. It can bind to a stretch of a protein and start the function there, which a small molecule not can do, right? And when we look backwards, I mean, there's one medications, insulin. I mean, since over 100 years now, this is a protein we use, and there has been no small molecule can replace the insulin. So we need also peptides to fight and cure diseases, especially when it comes. I mean, SARS is one of the examples where we got very fast a good vaccination. We have nothing found so far against AIDS. Cancer is still on the list, and the peptides, the small peptides, will be definitely on number one, but I see larger opportunities in the small molecules because they are more potent, they are more dedicated to the binding sites, they are much more specific, they are not toxic at all. So there is a bright future, even if it's just 5 or 10% of the market.
Well, it's a good point because macrocycles have this huge surge in research right now, and lots of companies looking at macrocycles where you're playing in that space between small molecule and peptides. And so I'm assuming you see lots of people working on macrocycles with the MultiPeps.
There's a few groups in Europe working on this, on cyclized peptides. We get also, I mean, for all of our instrumentation, we get requests from customers. They want to have a protocol, or they want to have the method. How can we do the cyclization? How can we do this on the membrane? Can we do this on the resin? This is always a question, sure.
When we have to be aware that, of course, other technologies come up which partly substitute what spots can do. I mean, we just talked on the cyclide peptides screening, so there are very powerful phage-derived methods that present cyclide peptides, the bicycle ones, for example. And so I think spots still has its relevance if you go for chemistry that you cannot do in a biological context. And spots is really flexible what the chemistry is about, and you can adjust a quite broad scale of chemical reactions to this type of performance. So therefore, I'm not really aware that spots will be totally substituted by any other method.
Well, yeah, I guess I want to thank you both, because I think it's a great point that you made, Christian, of just getting awareness out there of these different techniques that are available to people. And yeah, Dr. Frank, I guess you've seen a big transformation since the first report in that EPS, was 21st EPS 30 years ago. So yeah, very exciting.
It's fun to see that so many research labs still use spots.
Right. I do have one final question that may or may not go in. We'll see. But I'm wondering, for maybe a graduate student who's really interested, and they say, I want to do spot synthesis, but they can't go, get their professor to buy an instrument. Can they just get the membrane and try it themselves? I understand the intensity of labor, but if it's only like 5 peptides for proof of concept, is that something that you've seen?
We have seen this in the past. I still have a written manual for manual synthesis and pipetting. We have the lovely picture of Ronald Frank's arm with he holding the pipette. So this could be the front page. So I'm happy to share the information and the manual with you. And in the beginning, I remember that Heinrich Gausepol did a seminar in Brazil I think in the late 80s or early 90s. This was covering the manual synthesis. And that's right. Generally, for an epitope, something between 12 and 15 amino acids is already good. And if you have a certain area where you see this could be a binding site, this can be done. It's not really hard work. It's a little bit tedious.
Great. Well, no, that's great to know.
Maybe I can add that there are some recent reports even where you can see from the picture of a spot membrane that these were made by hand, because they're not that ready. So there are still some people pipetting on paper.
All right. Well, again, thank you both so much. I appreciate your time and the information.
It was very kind.
It's been really fun. And Ronald, at this point, I can thank you. As a chemist, I would have never thought to go so deep into the biology, because with the spot synthesis, it is really fascinating to combine the chemistry and see the outcome in the biology. Thank you.
Thank you for tuning in to this episode of Exploration Science. For additional details on the topic, please see the description box for links to articles and other resources. If you enjoyed the interview, please like, share, and subscribe. As always, we welcome your feedback and suggestions, which you can leave in the comments section. Thanks again for tuning in.